Hi Year 11s, welcome to the continuation of the cells and multicellular organism topic. Today we're going to be looking specifically at the cell membrane. This is our fifth PowerPoint. This one may be, no, it's going to be one video, but it's going to be quite a long video. There's 20 slides that we need to look at. There is some drawing, there is quite a bit of writing, so let's begin. We will go through the need to know in class, as per usual to say standards. It's not in the video, but it will be in the PowerPoint in class, so make sure that you're taking those down and just remembering which of the criteria you need to remember. So first off, what exactly is the cell membrane itself? Well, the cell membrane is an 8 nanometer long or wide boundary that separates the cell from the extracellular environment. So basically it's a semi-permeable wall that's creating two different environments. You've got the inside cell environment and the outside cell environment. The purpose of that cell membrane is to control the movement of particles in and out of the cell. It does this because it's known as selectively permeable. That means that some materials can pass through the membrane, but others cannot. Part of the things that make the cell membrane selectively permeable is that it consists up of transport proteins, which are embedded in the protein layer, or the lipid layer, and we'll talk about that when we start drawing this cell membrane itself. Lipids make up around 50% of the mass of the cell membrane. There's three different types. You've got phospholipids, which make up 50-60% of the 50% mass, and then you've got cholesterol and glycolipids combined make up the remaining 40. So what exactly are those things we're talking about? So let's talk about phospholipids themselves. Phospholipids are molecules that contain a phosphate head, which is the red circle here, and two fatty acid chains, which are the yellow tails coming out. The phosphate group is referred to as hydrophilic. It loves water, while the lipid tails are known as hydrophobic, which means they hate water, they fear it. So what happens is with the cell membrane, there's nothing actually holding the phospholipids together. What happens is when they are exposed to water, the phosphate head will be attracted to water and move it on the outside, and the hydrophobic tails, the lipids, because they don't want anything to do with water, they'll move on the insides, and that causes two layers. So the water will be on the outside of the cell, around here, on the phosphate molecules, and it'll be on the inside of the cell, touching the phosphate molecules. But the tails themselves will have no water exposed to them. So that means here is a hydrophobic section of the cell membrane. And we can see that a little bit better in this drawing. It's not the best drawing, I do apologise. But it gets the point across. So we've got over here just our phospholipid molecule. You can see the circular phosphate head, which is hydrophilic. And you've got your lipid tails, which are hydrophobic. And that's one of those is one phospholipid molecule. And those are repeating units that make up the phospholipid bilayer. And you can see two layers. You can see the phosphate heads facing towards the extracellular fluid, and you can see the phosphate heads facing towards the intracellular fluid. The phosphate heads and the fluid on both sides are hydrophilic, they love water. Whereas the lipid tails on the inside are hydrophobic, they hate water. You've also got a number of different proteins and cholesterol. So for example, we've got cholesterol here. We'll talk about cholesterol in a little bit. Basically, the purpose of cholesterol is to make the cell membrane more rigid or less rigid depending on the situation. We've got integral proteins. Now, integral proteins are embedded throughout the entire protein, so they're integral to the structure. As you can see, it goes through both layers of the membrane, and sticking out on the top there is a glycoprotein, which we use for cell recognition. We'll talk about them a little bit in a minute too. We've got another type of protein here in green, which is the peripheral protein. This one doesn't go through the entire membrane. It's just like an add-on, and it can move around the cell membrane. So the integral protein can't really move around, but the peripheral protein can. That's part of the reason it's called peripheral. It's an add-on. Peripherals are add-ons. We have the channel protein over here. The channel protein is another type of integral protein, and as you can see here, it's got a hole which allows some particles to move through. So this is one of those transport proteins we were talking about before. That's responsible for making the membrane selectively permeable. That is basically everything you need to draw for now of the cell membrane. So you can pause the video and draw that if you need, but let's continue. Talking about how the membrane itself changes in temperatures. So at lower temperatures, the phospholipids, they'll pack together and they're really tight, and that means the membrane is less permeable. It's like if it's really cold, you generally find yourself sitting closer to the people around you to maintain body heat. And if that was the case, there's less space for people to walk in and out between you. Whereas on hotter days, and everyone's feeling a bit sweaty, they stand apart from each other, and hence there's a lot more room for things to move back and forth. So the cell membrane works the same way. In cold temperatures, the phospholipid molecules pack tightly together, less permeable. When they're hot, they separate further apart and the membrane becomes more permeable. Cholesterol. So we were talking about cholesterol. Cholesterol is another big part in controlling how permeable the membrane is. So there's molecules of cholesterol, which in this diagram is the little brown diagrams, little brown molecules here. It was pink in the diagram that you were drawing before. And they're sandwiched between the lipid tails in the membrane. What they're doing is they're restricting those tails' movements, preventing the phospholipids from moving too much. Therefore, when we don't want when the temperature is changing, for example, it's going to prevent them from becoming too solid in lower temperatures because it can't let them get too close, but it's also not going to get them too far apart when it's higher temperatures because it's going to hold them at a certain distance apart. So cholesterol is really important for helping control the rigidity of the cell membrane and how permeable it's going to be. Glycolipids, again, glycolipids is the one that we're seeing here, attached to the integral protein. So glycolipids, these are carbohydrates that combine with lipids to make a glycolipid. It basically just projects from the outside of the surface of the cell, and it helps the cell with cell recognition. It helps the cells recognize each other. It allows the immune system to say, yep, that's one of the cells from my body, let's not kill it. Other cells will have different glycolipid, and your body will recognize that as foreign, and it can send the immune system to destroy that cell. Proteins. So proteins are another big thing. So going back to the membrane itself, just quickly, this is what's known as the fluid mosaic model. It's fluid because the membranes can move around so freely, and it's mosaic because you've got proteins embedded randomly throughout it. So the two parts, the phospholipids and the proteins. We're now talking about the proteins. So major component of the cell membrane, you'll probably have 2 to 4 times the mass of lipids in proteins. Like I said before, it's more like a mosaic cell membrane because it's got so many proteins embedded in there, hundreds of different proteins that form clusters in the fluid region of the phospholipid bilayer. And the two that we're going to look at are integral and peripheral proteins. So integral and peripheral proteins. Integral proteins are the ones that are embedded entirely throughout the membrane. Basically, they're there. They allow us things like transport channels, allow molecules and water to travel through the membrane without being exposed to the hydrophobic sections of the cell membrane itself. Whereas peripheral proteins, like we said before, they're like the add-ons, like a keyboard or a mouse, a gaming peripheral, or your peripheral vision when you're driving. They're loosely connected to the lipid bilayer and they can move around. So integral proteins not moving around, peripheral proteins can move around the membrane. Most lipids and integral proteins are mobile. They'll move laterally within the lipid bilayer of the cell membrane, so back and forth along lines like that. And research has shown that around between 30 to 90% of all integral proteins are freely mobile. Many receptor proteins, they're mobile. They'll move through the lipid bilayer to search for signal molecules in the extracellular space to help the cell bond and connect to them. Immobile integral proteins are also available though, and what they do is they connect to the cytoskeleton of the cell and they help anchor the membrane to the cytoskeleton to help provide that structural support. So, continuing on with the types of proteins. There's 4 different types of protein, or 4 different functions that proteins do. So 1, there's transport proteins. They're going to transport hydrophilic solutes across the lipid bilayer, so we're talking things like channel proteins like you drew in your diagram. Receptor proteins, they're going to relay signals between the cell and the extracellular space, so like your glycolipids. Enzymes, less so in the cell membrane, but we see these throughout the cell in general. They help to catalyze chemical reactions, to speed up those chemical reactions and let them occur at a rate that supports life. And like I said before, cell recognition as well. Again, help you recognize other cells. So now that we've talked about the cell membrane, we do need to talk about basically how particles are moving across the cell membrane, and you are going to be doing an experiment on this. So simple diffusion is the simplest movement of particles across the membrane. Going back to year 9-10, talking about what diffusion is defined as the net movement of particles with the concentration gradient. So they're moving from high concentration gradient or high concentrations to low concentrations in order to balance out and reach an equilibrium, so you've got equal concentrations on both sides of the membrane. This is a passive process. That means it happens without energy. a common mistake is once we've reached equilibrium, people say there's no more diffusion, which is not true. There's no net movement, so they will try and remain as stable as possible at that point, but you're still going to have some molecules moving back and forth across, but they'll be swapped over. There'll still remain an equal amount of concentration on both sides, but you may have one or two particles move across, which alters basically what's on each side, but it's still the same concentration. Diffusion will depend on a number of things, so how thick the membrane is, the surface area of the membrane, type of membrane, or type of material the membrane is made out of. Lots of things will control the diffusion rate. Osmosis, on the other hand, osmosis is like diffusion, but for diffusion of water. So two different statements there. Water will move from low concentrations of solute to high concentrations of solute. So water moves from very dilute sections to highly concentrated sections in order to have an equal concentration on both sides. They could also be worded of whether the side of the cell that's got lots of water, the water will move out to the other side, so there's equal amounts of water on both sides. So osmosis, water moving from low concentrations of solute to high concentrations of solute, or in other words, from high concentrations of water to low concentrations of water. That in itself leads us to a topic called tonicity. So that's comparing the inside cell concentration to the outside cell concentration. And there's three different terms you need to know there. So we've got isotonic. Isotonic is when the cell membrane, or the inside of the cell and the outside of the cell, have equal concentrations. And then there's no change via osmosis because there's no need, or no net change, because there's no need to exchange water across the membrane because you've got equal concentration on both sides. Hypotonic, and the key here is hypo is low. Hypotonic has solute concentration outside the cell being lower than the solute concentration inside the cell. That means the cell is going to take in water in order to equal out the concentrations, and because it's taking in water, the cell will continue to swell up and get bigger and bigger. Now, that has the potential to cause the cell to burst if it continues to stay in a hypertonic solution too long. Hypertonic, on the other hand, is when the solute concentration outside the cell is higher than the solute concentration inside the cell, and this causes the water inside the cell to be pushed outside to help keep the concentrations. That causes, the water leaving the cell will cause the cell to shrink, potentially also shrivel up and die that way if it's left in the hypertonic solution. So ideally you want to be an isotonic solution, but we can also use hypotonic and hypertonic to get water to move in and out. Okay, continuing on. Facilitated diffusion. This is another passive process. So far simple diffusion and osmosis have been passive processes. That means they're not using energy. Facilitated diffusion is another type, because it's diffusion, of passive movement where we're not using energy. Consequently, that means that the particles are going to be moving from high concentrations to low concentrations. Facilitated diffusion does, though, require channel proteins and transport proteins because the particles are often too big to move across the cell membrane. So we need to have some channels or proteins to allow them to move through. Channel proteins themselves will have a hydrophilic core in the membrane, so those small polar molecules will be able to diffuse through the cell because they're never having to be exposed to that hydrophobic section of the lipid tails that will repel them. Channel proteins can be opened or closed to control how much of those particles are going to move across, and they're also specific to specific ions. So for example, you might have a sodium channel or a potassium channel that's only going to let sodium or potassium come through. We also see them help move substances like glucose and amino acid because they're too big to come into the cell through diffusion or osmosis. Again, these will have specific binding sites that will trigger their entrance into the cell. Once the substance will bind to the protein, the tertiary structure of the protein, so the actual shape of the protein, will change and it allows the molecule to move across the membrane, and then the transport protein will return to its original shape. Again, there's a number of factors that will affect the rate of facilitated diffusion. Availability of proteins is the biggest one, because if there's not enough proteins, your diffusion is going to go very, very slow. Active transport, on the other hand, active transport is moving particles from areas of low concentration to high concentration. So this is against the concentration gradient. Particles want to move away from other particles and have an equal amount of space. Active transport is the opposite. You're forcing, so this one over here, to come out into the extracellular fluid. Now normally what we'd expect to happen in diffusion is we'd expect 3 of these to come over to this side. But active transport means we're moving this last molecule out there, even though it doesn't want to. To do that, we need energy, which you can see over here. ATPs help powering it, and we're using carrier proteins here that are going to help push that protein out. So ATP will come along, bind to the protein, cause the shape to change. This particle will come and land inside the protein itself. The energy will leave, the cell will change back, and it will force that molecule outside the cell, and won't be able to get back in unless energy's open. So 2 different types of active transport. These are also called bulk transport, is endocytosis and exocytosis. You could sort of figure out what those mean by the name. Endocytosis is bringing something into the cell. So basically what's happening here, because the membrane is so fluid, the phospholipids can move around, pinch off and pinch out of the cell and change shape. So small amounts of membrane can be added or removed from the membrane, is basically what we're saying there. Endocytosis is taking up materials into the cell, so a solid particle will be engulfed by the membrane, so the membrane will pinch out and kind of grab it, and that part of the membrane will move inside, forming a vesicle, carrying that substance from outside the cell into the cell. This is how organisms will feed. There's a process called phagocytosis. Phagocytosis is engulfing of solid particles, whereas pinocytosis is basically fluid particles and they're drinking. So phagocytosis is like the cell eating, pinocytosis is like the cell drinking. They're not actually eating or drinking, but the processes are very similar. Exocytosis, on the other hand, exo meaning outside. This is when we're using active transport to basically remove a large amount of molecules from the cell and push them, also they're coming from inside the cell and we're taking them outside the cell. Like I said, another form of bulk transport. Almost exact opposite of endocytosis. You have a number of particles inside the cell surrounded by a cell membrane. Or by a membrane, not a cell membrane. That membrane will move up and fuse as part of the cell membrane, and eventually the cell pinch itself out and those particles will be forced out of the cell. So you can see here the vesicle moves up, fuses with the membrane, they kind of mix together to the point there's no vesicle anymore, and the particles are left on the outside of the cell. Surface area to volume is a big part of this, and this is what we're going to be doing a prac on. I'm not going to go through these 2 slides because there is another video on surface area to volume ratio, which is on my tasks for homework. So if you watch those videos, that will explain what surface area to volume ratio is, and we'll also do an experiment on this idea of surface area to volume ratio with some agar cubes, and that'll be coming up in a lesson very soon. I will stay on these slides, or you can feel free to pause these 2 slides if you'd like to take that information down, but otherwise I will see you in class.